I'm going to talk about a paper that's fairly exciting that was just published this month, November of 2011, and it was published by several departments of the Mayo Clinic in Rochester, Minnesota, as well as between a collaboration with Groningen University in the Netherlands. I might have pronounced Groningen wrong. Yeah, and just a little bit about the Mayo Clinic. They're quite a good clinic and hospital. They have several different facilities. Their headquarters are in Rochester, Minnesota, but they also have a Mayo Clinic in the Phoenix, Scottsdale, Arizona area, and then also in Jacksonville, Florida. And the Mayo Clinic has been rated among the best healthcare places in the world, and they also do a lot of combination between treating patients but then also core research too. They try to put 40% of their effort about into research. So in this paper, it's about the aging effects of senescent cells, and they were able to remove the senescent cells from mice and show that this reduced age-related phenotypes. The mice were much healthier. Yeah, before I go too much further, I just want to talk about one sentence they have here in the abstract. They say cellular senescence, which halts the proliferation of damaged or dysfunctional cells, is an important mechanism to constrain the malignant tumor progression of tumor cells. So I mean, yeah, they claim, as well as many people claim, that the purpose of cells becoming senescent is almost to prevent cancer. And as I'm sure many of you know, a cell will become senescent after its telomeres get too short. So cells can keep on dividing, and every time they divide, their telomeres will get shorter and shorter. And once those telomeres reach a critically short length, then the cell will either no longer divide and become senescent, or it could escape this stopping of division, and they can become senescent. So, but I just wanted to say that I don't know if this necessarily has to be the case. Another theory that some people have proposed for the purpose of cellular senescence is maybe it's not really so much to stop cancer. Another possibility could be that cells just become senescent because there was never any reason in the past for evolution to design an organism like ourselves that had cells that didn't reach senescence. So the organism just had to live healthy long enough to reproduce in its environment, and if it reached that goal, then there's no really pressure to escape this senescence mechanism. So rather than having a purpose to stop cancer, it may have just been the avoidance of senescence may have just been left out because there wasn't evolutionary pressure for it. Anyway, enough of that. I'll go on and start talking about the paper a little bit more here. So the mice they were working with, they were boob R1 progeroid mouse background, and some of you may know about the disease progeria. There's some humans that have progeria as well, and when they have progeria, they age extremely quickly. Even so, when they're teenagers or in their early 20s, they look much, much older, and they die at a young age. And so in this particular mouse, they have something similar, and they have a mutation that causes the chromosomes to not separate properly during cell division, and so this causes problems and more rapid aging. And some of the cell types and tissues that are most affected by this process are the adipose tissue, so the fat tissue and skeletal muscle and eye tissue. But then some other things that won't be as much affected by this process of reaching senescence would be things like heart cells, which divide very rarely, if at all. So that's something to keep in mind as they go into the results later in the paper. Okay, so right now I'll just show you the schematic of this construct that they put into these progeria mice. This is the ink attack construct, and so they genetically engineered their mice in such a way that when they administered a drug, they could kill just the senescent cells. So kind of the way it worked here is they had they had this P16 promoter. So P16 is a biomarker in some senescent cells. So it's a gene that is turned on in senescent cells. And then so this gene would normally turn on and produce P16, but then they also added these other genes that would be transcribed or created with it. So one of the genes is caspase, and then the other one was green fluorescent protein, this EGFP. So the green fluorescent protein would just allow them to detect which cells had this genetically engineered construct. And then with the caspase, so what that's all about is actually caspase is turned on when a cell undergoes apoptosis and kind of kills itself. In fact, when a T cell of the immune system finds a cell that needs to kill, what it will do is it will activate some pathways which will eventually turn on caspase, and then caspase will tell the cell to undergo apoptosis and kill itself. So here, with the way this system works that they created, the caspase will be created, and then it won't have any effect until they add this drug, which is AP20187. So this AP drug. And once that AP drug binds, it turns this inactive caspase into an active caspase, and the cell will undergo apoptosis. So now all these other figures here, I'm not going to spend time talking about. All they're doing is they're showing that their construct does work the way that they expect it to work. And so yeah, here's a sentence summarizing all those figures. Taken together, these results indicate that ink attack, which is that genetic construct, is selectively expressed in P16 positive senescent cells. So senescent cells will produce that P16. Okay, so now let's get to the results with the mice and see what happens there. So first here they show that when they do add the drug, it does truly kill these cells that have been genetically engineered to respond to this AP drug. So this drug, rosiglitazone, what it will do is it will cause cells to become senescent and express those senescent biomarkers such as P16. And then here in this figure they do not add the minus AP. But then, this is a little bit of a fuzzy image, but here they do add the AP, and you can see there's much fewer cells here. So it is true that the AP does kill the cells. So now for these mice, they had different groups of mice, some which received the AP drug throughout their lifetime, so the senescent cells were being killed and removed, and some didn't. And the ones that received the drug fared better. So here, what they have on this curve is they have the percent of the mice which had lordokyphosis, which is just like a curvature of the spine associated more with old age and damaged skeletal muscle tissue. And with these dotted lines, you can see that they had a much lower incidence of lordokyphosis. And the dotted lines up here are the ones that received the AP drug, so plus AP and plus AP. You'll see this is attack 3, this is attack 5. They're just two different cell lines that both have this genetically engineered construct. And then the minus AP ones here are these dark lines, and this didn't even have the genetically engineered construct. And you can see these lines here, they have a much higher rate of lordokyphosis. They also measured cataract incidence, and same thing here, there was a lower incidence of cataract with the mice that had these senescent cells killed by the administration of the AP. So that's quite interesting. Here's actually a screenshot comparing the two mice. So here's the ones that received the drug and the senescent cells were killed. You can see they look almost kind of chunkier, but their posture looks better. They don't have that curvature of their spine. They look kind of healthy. Whereas the and these are 9-month-old mice. These particular progeria mice, if I recall, they just live a little over a year, not too much longer than that. And then these mice, they did not receive the drug and died sooner. So they also show the muscle fiber diameters are thicker. So here, the light blue and the orange received the drug, and they have thicker muscle fibers in both the muscle associated with the stomach area, or the gastrointestinal area, and then also the abdominal muscle. And they also compare treadmill exercise. So on this graph, both there's the 3 and the 5 lines of mice, and this is the percent improvement over the untreated mice. So these mice could run on a treadmill for a longer amount of time, further distance. They performed more work, also, like work as you would calculate in physics. So it's quite impressive. They also measured the cell diameter of fat cells and the thickness, and the fat cells and thickness were higher were larger, greater, in the mice that received the drug and had the senescent cells killed. This here, they just have a table of numbers that's kind of showing the same thing, and the mass of these cells was measured with a technique called DEXA. I'm not very familiar with that, but DEXA is, here I made a note down here. DEXA is dual energy X-ray absorptiometry. Yeah, so that's kind of interesting. This figure over here, I won't spend much time on that, but I do want to talk about the next figure, because so everything I talked about before, they were treating the mice from a young age. They kept giving them that AP drug, but they also wanted to see what kind of effects they could observe if they started a late treatment. So the mice were already kind of old, and then they started administering the drug. And basically what they found, maybe I won't even go into too many of the details of the figure, but I'll just summarize what they did find here. And they measured many of the same things they did before. For example, muscle fiber diameter, how far and how well they could run on a treadmill, the size of fat cells, all those different things, as well as the relative expression of different genes associated with senescence. Mice that receive the treatment reflect attenuated progression of age-related declines rather than a reversal of aging. So basically, they weren't able to reverse the aging as well. On these figures, they show 10-month-old mice, and then they also just compare that with a 5-month-old mice, the black bar. The black bar always has the best value, so the 5-month-old mice always have the best value. So when they started the treatment at 10 months, they weren't able to reverse the condition of the tissues back to the healthy state of a 5-month-old mice, but it was better than the mice that received no treatment at all. So basically what they were able to achieve is attenuation of these age-related phenotypes rather than reversal. So let me think if there's anything else I want to point out here. Importantly, they didn't notice any overt side effects, so that's very positive. And also, so I'd just like to spend a little bit of time talking about what we would need to do in order to transfer this treatment to humans. So basically, in order to apply this treatment to humans, you wouldn't be able to copy exactly what they did in this paper, obviously, because we haven't been genetically engineered to respond to this AP drug and have our cells be killed when that drug is administered. However, what scientists could try to do is identify specific biomarkers unique to senescent cells. For example, even these two papers go into, which I haven't read yet myself, but they go into more details about specific components that senescent cells secrete or possess. And you can try to develop drugs or specific therapies that will target and kill cells that have those things. So it would take some work to figure out exactly how to do that, but theoretically it would be possible, and then you would also be able to delay age-related phenotypes in humans, so we could be much older in old age, much healthier in old age. And another question about these mice that many people would ask is whether or not they actually lived longer, and the answer was no. So these mice that received the AP drug and had the senescent cells killed did not live longer, and the reason was they appear to have died from cardiac failure or heart failure, and so all the problems that you'd have with a normal heart or in the control mice, you also saw in those mice, because those were tissues and cells that were not affected as much by this senescence process, because there's not a lot of cell division in the heart anyway, so they still had the hardening of the arteries and everything. So if you did want to treat a human and prolong the life, you'd also need to develop treatments that can help these tissues that don't have a lot of dividing cells, such as the heart. I believe also the brain does not have a lot of cell division. There's one last comment that I'd like to make, and that's about a company named TA Sciences in New York. And TA Sciences, TA is for telomerase activation. This telomerase activation science company, they're kind of a branch off from the Geron Corporation, which is a big biotech company. And what they did was they just screened many different compounds from different areas of nature, and they identified this one compound that came from a Chinese tree that was capable of activating telomerase. And so this telomerase enzyme, once it's activated in your cells, it will lengthen the telomeres and prevent those cells from reaching senescence. So this company is actually selling this compound to people today. I think it's kind of expensive, but they are selling it to people today, and there's people taking the drug. And the company has also had academic third parties publish academic papers testing their compound. And from what I've seen, I think it looks legitimate. I mean, you can never be completely sure. I mean, there can be a lot of fraud in the world and everything. But after this paper that was published this month about removing the senescent cells was published, TA Sciences also sent out an email just describing how it's great that removing the senescent cells did show such benefits. However, their treatment is focused more on making sure that the cells never reach senescence in the first place. And so that may, and they claim and have tested and seem to have shown that has a lot of great benefits as well. So possibly in a human, if you could do both, if you could make sure that fewer cells even reach senescence and then also remove the ones that do become senescent, then that may be a pretty good treatment and increase the health span of people.